What's good, class, and welcome back to another edition of Mr. Fitzgerald's biology class. I know what you're thinking already. Did he sleep in those clothes? Absolutely not. I recorded 2 videos in one day because I'm getting smarter like that. But anyway, today we're going to dive deeper into the cells and look at cell organelle part 2. So you can add these to your cell organelle notes from the previous day and continue them on, or you can start a new page because we do have some new fascinating parts of the cell to explore today as well. All right, so let's go right into it. Kind of start you off with a little bit of a question. Think about it. Why do you think the endoplasmic reticulum is positioned right here next to the nucleus? All right, so considering what we learned yesterday with the nucleus and the endoplasmic reticulum, remember the nucleus is the location where DNA is stored inside of eukaryotic cells for the most part, and that DNA has the instructions on how to make proteins. The endoplasmic reticulum, if you recall, is the position where proteins are made for the most part because it has these structures embedded on it called ribosomes, and ribosomes build the proteins. So I like to think of the endoplasmic reticulum as the factory and the ribosomes as the factory workers because they're the ones that are actually building it. And the endoplasmic reticulum is just a nice location for them to do their job. So instructions come out of the nucleus and go right into where they need to be built at the factory to where the workers, the ribosomes, can actually build them. So the position of the ER is always right next to the nucleus, so that way the proteins can be manufactured in a much quicker and more efficient process for the cell. All right, so let's talk about these ribosome things, okay? So what was their job again? Their job, remember, is to make proteins, okay? They are actually made out of protein as well, and a structure called RNA, which we're gonna cover a little bit later. And they're also created inside of a very special region of the nucleus called the nucleolus, all right? Now, do I expect you to fully remember that word? Kind of. Know that the nucleolus is an area inside the nucleus where ribosomes are made, and that's about it. It's not going to be covered too much more in detail by me because it doesn't usually come up a lot on questions. They focus more on protein production, which is more of the nucleus as a whole, and then the ribosome doing the actual job from those instructions. All right, but ribosomes are your protein makers, so they are going to be the ones that are influential in taking those instructions from the nucleus, the genes, the DNA, all that stuff that holds the information, and they're the ones that actually implement the protein production and actually make what the cell needs at the time. All right, so where can we find some dots? Now, the reason I say look for the dots is because that's what ribosomes are usually shown as in pictures. If you did your types of cells activity, you actually noticed that there were some dots in those prokaryotic cells as well as the eukaryotic cells, and they had them labeled as ribosomes. Same thing here, look for dots. We know that the endoplasmic reticulum, the rough ER, is gonna have dots on it because we have ribosomes embedded on the rough ER, making it called the rough ER. We also see that there's a lot of dots throughout the cytoplasm. So there are some proteins that are not just made at the factory, they're made outside. And so the instructions get moved into the cell plasma area, the cytoplasm, and then they're able to use those instructions to build the protein. So you will have protein production happening within the cytoplasm as well as inside the endoplasmic reticulum. So the 2 locations where you can find ribosomes and protein production is going to be free floating in the cytoplasm or directly connected to the rough ER. So protein production can happen at those spots. All right, so let's talk about some other ones instead of kind of reviewing things we've known and just adding little pieces to them. So let's talk about some more fancy ones. This is called the Golgi apparatus, or sometimes it's called the Golgi complex. And to make it more confusing, sometimes they call it the Golgi body. So really the word you're looking for is Golgi something or other. All right, now, what is this thing? It always kind of looks like just a squeezed together sack of something or other. And they're basically flattened sacks of membranes that are basically the packaging and last processing area for the proteins that are produced in the ER, or the carbohydrates or the lipids that are produced in that endoplasmic reticulum, either the rough or the smooth ER, and then they can get moved throughout the cell where they need to go. So sometimes they like to actually add in some pieces to it to kind of finish that process of building it. Maybe it just needed some refinement or some extra characteristic added. So that can be done at that Golgi body. And then it ships it off to where it needs to go. Also, what's cool is this is usually where you kind of get some of the trash taken out of the cell. So sometimes I said last time, the cell builds proteins or carbohydrates or lipids just because it has extra stuff or it has waste stuff, some surplus of something, and it puts together all that surplus. And the Golgi then recognizes, hey, this is a waste product, and actually sends that vesicle or package all the way to the cell membrane to actually expel it out. So parts of this will break off in order to send those packages to where they need to go in the cell. So I like to think of this as the Amazon of the cell. It has so much stuff it contains, it modifies, puts them in better boxes, and then it ships them out to wherever they need to go. All right. Now, vacuoles are another interesting thing that we can run into, and depending on if you're talking about a plant or an animal, they kind of get used differently. But vacuoles are usually used for storage of food or maybe an enzyme or another material that's needed by the cell. Usually in plants, the vacuoles are much bigger, right? You get this large area here, whereas in animals, they're usually short-lived. They don't hold them for very long, and they typically are very small when they happen. And so inside of an animal cell, it's because we don't store our food in our cells for very long. If we have a little bit of surplus, sometimes we store it up, but we basically are burning through anything we need very quickly. Whereas plants actually have to pay attention to water concentrations, they have to pay attention to the amount of food that they're producing because sunlight is not consistent year round. So plants aren't able to always produce food at all times. So then some of the times the vacuole is used to store up food. Sugar in this case that they're producing from photosynthesis. Or maybe there's droughts. Sometimes it gets dry, especially in Florida. We go days of rain and then days of no rain. So this can actually store up water for the cell, so it can still do some photosynthetic processes, still can do the things that it needs to inside of itself, but it doesn't have to rely on the roots pulling up water that isn't there at the time. All right, so why is it bigger in plants than animals? Again, plants sometimes need to store things for longer because they can't just go get it. Animal cells usually are mobile, so they can go get what they need if they need it. All right. Now, the lysosome, this is usually more of an animal cell characteristic, and it's kind of a cool thing. So this is a membrane-bound organelle that has really strong enzymes in it to break basically everything apart, from food particles to bacteria to viruses to the cell itself. There is a process called apoptosis, where it's called programmed cell death, where basically a signal gets put out by the nucleus to say, hey, it's our time as the cell, and they can actually burst all these lysosomes throughout it and destroy the cell from the inside out. That's usually used in cases for us, like infection or something like that. We'll lose a couple 100,000 to millions of cells. Don't worry, we can replenish them pretty quickly because our cells usually reproduce every 24 to 48 hours, so most of our cells get reproduced very quickly. Also, you have over 100 trillion cells, so you'll be fine losing a couple million. So it is so important for this one to have a membrane because those enzymes are so aggressive, they will literally eat the cell inside out. So if the cell doesn't protect these lysosomes very well, unfortunately it's going to end up poorly for that cell. And if it's a single-celled organism, there's nothing to replace it. But you and I, or a plant, or whomever would actually have to go through that process, we have tons of cells, so if we had to lose a couple of them for reasons of it was infected by a virus and it's beneficial for the organism, the cell will actually go for itself. All right, now we start getting into some of the more complicated parts, okay, the cytoskeleton. Just like you and I have bones for a skeleton, cells have a skeleton-like structure, but it's made out of proteins. Okay, so this is a large network that kind of supports the cell, gives it anchoring ability, gives it the ability also to move and transport materials as well. So it's basically just kind of a network of maybe bridges or arches or supports and other things so the cell doesn't collapse upon itself. They get fairly large, so you can imagine that it would be hard to hold that cell shape that it has without having some internal structure. So the cytoskeleton really provides a lot of internal structure in the form of proteins. Okay? Now there are two parts to the cytoskeleton that you kind of do need to know. They're called microfilaments and microtubules. Okay? So the microtubules are hollow tubes that allow things to kind of move around in the cell, more so than providing the ability for the cell to move as well. So they provide a network of kind of highways for materials to move, as well as providing large support structures for the cell to prevent it from actually collapsing as well. On the other hand, microfilaments are a little thinner, so they're more minute, and so this gives the flexibility for the cell to not only maintain its shape, but also to kind of move and adjust. So the tubules are a little bit more structure-wise. The filaments are a little bit more fine, so they can kind of move around a little bit and get some light movement. So cells that are mobile have a good number of microfilaments to allow their flexibility within their cytoskeletons. All right, that's all we have for our cell organelle journey. We will actually talk about some plant-specific organelles in the future as well, because remember, plants actually have more complicated cells than animals. But make sure you rewatch this video. I know I went through a couple of things pretty quickly to kind of gain all that information and knowledge, and make sure that you have it all written down in your notes. That's all I have for today, and have a great one.